my white blood cells, macrophages, are going to start to release interleukin-1. TNF also does the same thing. It triggers a change in the set point. So even though my temperature right now is 98.6, now my set point is now, let's say, 102. Temperature 98.6, pyrogens have changed my set point to 102, fever. How will your body respond when now how is your body temperature at this point compared to your set point? Is your body temperature the same as your set point, above it, or below it?
Below.
It's below your set point, correct?
Yeah.
And how does your body respond when your temperature is below its set point?
It increases to get to the set point.
I'm sorry, what?
It increases
How
to
will
get
it
to the
try
set
to
point.
get your temperature to your new set point?
Heats up. Our body heats up.
Oh, okay. You don't understand. Okay. Well, here's what happens. When you're colder than your set point, you shiver. Nobody understands that. Yeah, when you're colder than your set point, you shiver. S-H-I-V-E-R. So you'll begin to shiver because you are what? Colder than your set point. And when you're colder than your set point, your body's response is to do what? Make you shiver. And now that my temperature is the same as my set point, while you are shivering, you will feel like you are what? Cold. You're going to feel, oh, I'm cold because you're lower than your set point, all right? You'll feel like you're cold, you'll bundle up, right? And yet you're going to be hot to the touch. Now let's say that you have fought the good fight. We're going to jump to the end of your infection. You have fought off the good fight, and now the level of pyrogens is dropping. So at this moment now, that your pyrogens are dropping, which means your set point is now coming back down to what? To normal value here. At this point in time, now how does your body's temperature compare to your set point? Is your temperature the same as your set point, above it, or below it? New set point here, temperature here. How is your temperature compared to your set point? So 98.6 is now higher than 102?
Wait, what?
I'm asking how your temperature is compared to your set point.
The temperature is higher.
Your temperature is higher, so how will your body respond?
You sweat? You sweat it out?
You will do what?
Sweat it out.
Sweat, because that's what happens when your temperature is higher than your set point. So you'll begin to do what? Sweat it out, and we say the fever has broken. Is this something that's usually a good sign?
Yeah.
Yes, that's usually. Now we're exaggerating a little bit, like you only get one round of this, and that's the end of it here. The values of interleukins and the pyrogens generally are varying while you're getting sick, which is why you usually often get repeated rounds of sweating and shivering. Nobody else has had this condition? Okay. But again, the bottom line is that fever affects what part of the brain?
Thalamus.
Hypothalamus, because that's where your set point is. So now we get to move on to true immune system response, adaptive immunity, that is different from all those non-specific defenses for a wide variety of reasons. One thing that it has is specificity, which means that you're going to only activate very specific lymphocytes to fight this specific infection. There's a massive amount of versatility because you produce hundreds of thousands, if not millions of different types of lymphocytes that can recognize just almost an unlimited range of foreign types of antigens. a big difference between this and the innate responses is that this has memory. Memory means that we're going to be producing these specific memory cells after infection that become active in response to secondary infections, a secondary exposure that can be months, years later. And it's the memory that will give you quote unquote immunity to it. And another property is tolerance. The lymphocytes don't respond to all antigens, just to their one antigen. Just tell them I'm not here. So this adaptive immunity is not something that you are born with. It gets developed. We have active and passive immunity. And I might as well mention this right now. We'll talk about, yeah, it's good to do this right now. Because I've got this slide up here. Let me see if I can make this a little bigger. Good time to mention this right now. The key thing about what's called active immunity is memory cells. The memory cells that have been produced during a primary response is what gives you active immunity. Now, there are two general ways of getting active immunity. Naturally acquired means that you got sick. You developed a primary response, you came down with polio. The problem is you will develop an immune system response, you will develop immunity to it. The problem is what happens to you during that primary response. You're getting sick. Many people with polio end up paralyzed or dead because of that. But on the other hand, if you want to avoid the whole death thing, you can get artificially induced active immunity, which is what vaccinations do. Vaccinations give you active immunity because they will produce memory cells without having all those nasty death-inducing side effects that happen when you actually get infected with a live pathogen. But the memory cells don't know any better, and they will be the ones that will then confer immunity to you so that you don't come down with smallpox, diphtheria, whooping cough, measles, mumps, everything you've been immunized, hopefully COVID-19. This is artificially induced, but it's still active immunity. So whether you get sick or get vaccinated, you've got active immunity. Now, passive immunity is very short-lived. Passive immunity occurs when you are protected by antibodies that you did not produce yourself. In passive immunity, you are protected by antibodies that you did not produce yourself.
So is it like a newborn baby? Oh. I'm just protected by what?
You are protected by antibodies that you did not produce yourself. And this is relatively short-lived immunity. It's only good for a couple of weeks, maybe a month, because the antibodies that you have been gifted are proteins that eventually break down over time. So passive immunity is very short-lived, where active immunity is years and years and years. So what do we mean by protected by antibodies that you did not produce? Well, one group of antibodies are ones that can cross the placenta from mother to child. The only antibodies that cannot cross the placenta are the weird ones for the ABO blood types. That's why you could carry a B baby. If you have A-type blood, no issues there. But every other thing that mom has been exposed to and developed memory cells for, and is producing antibodies, can cross the placenta, and that baby, when it's born, will have antibodies in all of their bodily fluids that mom has produced, giving that baby some immunity. Now, mother's milk also contains antibodies, a type that's called IgA that's found in secretions that mostly just stop the pathogens from being able to stick to your bodily cells, gain access, so that as long as a baby is breastfeeding, that baby will be getting some passive immunity passed on through breast milk. Now that type of antibody, is not going to get into circulation into the baby's body here. It's drunk with milk. It's going to be coating the digestive system, protecting mostly the upper respiratory and digestive, but still producing some immunity. The other place where you find passive immunity is artificially induced is in anti-venom serums. Anti-venom serums. What do I mean by that? Well, let's say that you're taking a nice little walk through the American Southwest and you get bit by a rattlesnake. Right? Don't you hate when that happens? Really, just me, okay. Now, will your body eventually be able to mount an immune system response against that foreign venom? Everybody should say yes, all right. But could you be dead by the time it does that? Yes, because it takes how long? Like a week to even develop that thing, all right? And you're gonna be 6 feet underground by that time here, all right? So you don't have time to develop your own immune system response against that. Well, the way this works is that somebody at some rattlesnake ranch, they will harvest little bits of venom like this, and then they inject a tiny little droplet of it. Just a teeny little droplet of it, like half a drop of it, into what they used to do, into a horse. And just a teeny, not enough obviously to kill the horse there, but enough to actually get that horse to develop what? An immune system response against it. Now, the horse will then develop an immune system response against it, will develop a primary immune system response. But is the primary response going to be as strong as the secondary response? Secondary response is always going to be stronger. So what they do, they give it a little bit, the horse develops a primary response, develops some antibodies, but then they wait a month or so later and give that horse another teeny tiny little droplet. And then that horse will produce a secondary response with antibody levels that are hundreds, thousands of times higher than they were in a primary response. If we're producing antibodies at 100,000 times, now we have enough that we can harvest. So they will take samples of they'll take horse's blood out, all right? They'll centrifuge it, they'll separate out the antibodies from it, return the blood back to the horsey, so the horsey can do the same thing again here, and they'll keep that little bottle of antibodies that have been made against the rattlesnake venom in a refrigerator someplace in some doctor's office, emergency room. So if you get bit by a rattlesnake, all right, that anti-serum, they will just inject you with a big bolus full of antibodies that have been made by, in this case, some sort of laboratory animal. I think they used to use horses for this. So again, is this going to be a long-lived or a short-lived
Sure.
protection? Short-lived, but that's all you need for this. Okay, so let's take a look at the adaptive immunity, the lymphocytes. They are the star player. You're already familiar with the fact that they're the second most abundant white blood cell in circulation. And they're going to include the T and the B cells. The NK cells we touched on earlier when we did the nonspecific defenses, because they are really a nonspecific defense. They are very long-lived. They can live years and years, 20 years or more, so they're not like neutrophils that live like half a day, or red blood cells that only last maybe 3 to 4 months. Much longer period of life. And again, they're not resident cells. They crawl around. They are visitors. They crawl around through the lymphatic system, move around. I don't care that you know there's a difference in speed between the T and the B cells, not an issue here. But lymphopoiesis occurs in multiple places, not just the bone marrow, but also thymus. You can get some lymphopoiesis in the spleen and other types of peripheral tissues. Once the stem cells have produced the lymphoblast, for example, they go all over and then finish reproducing here. Now this is showing sort of an overview here. Let me bring up a major difference between the T cells. The ones that really have killing function are going to be the cytotoxic T cells. We're going to compare those with the B cells. Because the cytotoxic T cells are best for intracellular parasites or intracellular infections. That's why they have to have things presented to them that were once inside the cell, because they specialize in intracellular infections. Cells that have been infected with viruses, cells that have been infected by bacteria. The cytotoxic T cells specialize in intracellular. Whereas the B cells tend to specialize in things that are extracellular, outside of cells, which is why B cells did not need to be activated by an antigen-presenting cell. They can be initially activated by just a free-floating antigen, like that little bit of cobra venom that we spoke about. No, it wasn't cobra, it was rattlesnake venom, right? Wrong country. And the B cells, again, are one-trick pony. When they get fully activated, they mature into this very bloated antibody factory that they just call a plasma cell. Plasma cell is just this big bloated antibody factory here that's filled with rough ER. Why rough ER? Why would they fill with so much rough ER? Remember, rough ER is where we did protein modification, right? The ribosomes on the surface where we initially do the production, inside we modify them. And so because antibodies are protein, we just fill this bloated big cell with rough ER, which is what they're trying to show here. And so once they get activated, they're producing thousands of antibody molecules every second. Just spin it out there. So B cells specialize in extracellular. The cytotoxic T cells are going to specialize for things that are intracellular. Now the other types of cells, we have memory cells on all types here, but the helper and the regulatory ones, the helper T cell is going to be critical for triggering cell division, fully activating both the T and the B cells here. Where the regulatory ones are going to tend to turn down the immune system, let's moderate a little bit more. The helper T's turn up the heat, regulatory T's, they turn it down. So I'm not going to ask any more about the hemocytoblast because we've covered that already before. We've already mentioned, so I'm not going to test you on that again. You might see it on departmental final at the end of the semester again, but we've talked about the stem cells that produce all the different types of formed elements here. We've talked about how the thymus gland that produces thymosin, thymopentins, thymopoietins are involved in maturing the T cell response here. And again, the cytotoxic T is the only T cell that can directly attack. The name itself, cytotoxic, tells you that much. The helper and the T and the regulatories, they can't attack. They can't kill anything. They just regulate the immune system response. Helper T cell coordinates it, brings it up. Regulatory T cells generally moderate, bring it down. So let's take a look at how this works. So adaptive immunity forms here, cell versus antibody-mediated, cell-mediated T lymphocytes. So this can eliminate pathogens that reside inside host cell, things that are endogenous inside of them here, because that is a location where they would not be accessible to antibodies. Again, cell-mediated response for the T cells for intracellular issues, where the B lymphocytes produce antibodies that are good for extracellular threats. Because those antibodies, the antibodies that have been produced by this will end up in all of your bodily fluids. Will you find antibodies in all your extracellular fluids, everybody? In plasma, in tears, in saliva, in milk. All of those secretions are going to sort of have those, which is why they often refer to the response as being anti-mediated or humoral immunity. Did we have the talk already about that term humoral? I thought we did, no? Yes?
I feel like we did once. Maybe
What does that term humors refer to?
Well,
No. Okay, all right, well, we'll have a short lesson. I thought we did this before, but we'll have a short lesson and a little bit of history of medicine. Since you folks, all right. This term humors refers to body fluids. Fluids. Did you come across any of these, any humors in 109?
no, we didn't. I don't know that. Yes.
Is that a sign you're giving us?
It's an extremely bad clue
I just want to say
I'm
yes.
trying to give you. Maybe something about the eyes.
Oh.
That wasn't what we were getting.
You were someplace completely different, weren't you there? Someplace completely different.
I thought you were waiting
That's
for
so
like a
wrong.
yes.
That is so wrong.
What?
It's like, what is he doing?
No.
So, but before they come to arrest me, in the eyes, the front part of your eye, the anterior chambers, were filled with a watery fluid that was called aqueous humor. Remember that?
Oh my God.
Aqueous humor. Aqueous humor was a fluid that helped to maintain intraocular pressure, keep your eyeball slightly inflated. All right? Because if it was inflated too much, high intraocular pressure would occur if there was too much aqueous humor, and that would start to kill off the optic nerve, causing a condition that's called glaucoma. Do you remember that at all now, glaucoma? Glaucoma from too much aqueous humor?
Yeah.
All right. Now it's coming back. So that term humor is referred to
causing them that.
fluids. And that's why they would refer to it as humoral immunity. If you go back to the days of Aristotle, I'll just kill a few seconds here. One of the older thoughts about medicine was that disease was caused by an imbalance of the 4 bodily humors. No? Didn't touch on this in here? So they used to think that diseases, and this goes back a couple, 2,000 years or more, that was caused by an imbalance of the 4 bodily humors. There was blood, there was phlegm, there was yellow bile, and then there was this mysterious stuff they called black bile, which I never understood. I don't think anybody understood, but they used to think that diseases were caused by an imbalance of this. And this was an early attempt at a scientific explanation for disease. Because if you go back to the day, if you look at causes of disease, you started with 2 camps of thoughts about why people were getting sick. You had the supernatural group, and then those that thought that there were natural explanations. The supernatural ones would say that you're getting sick, why?
You're possessed.
Like
You're what?
You're possessed.
You're possessed, or maybe a witch has cursed you or cast a spell, or maybe just God himself or herself was punishing you, you know, for that thing you did.
They don't like you.
Or that thing your parents did, right? Or there's some demon activity, there's something here that is causing sickness here. And there was a lot. And the natural, you had one of the earlier natural ones, it had some scientific background to it. It might not seem like it, but when people get sick often, it seems like, for example, they're doing things that they don't do when they're normal. For example, a lot of coughing, a lot of sneezing, a lot of spitting. And what are you losing? Phlegm, all right? So we're losing a lot of that. Maybe your other fluids just aren't in balance, or maybe you're throwing up a lot when you're nauseous, which means you're losing bile. Or maybe you're hemorrhaging, which means you're losing blood. Or maybe, I still don't know what the black bile was. It's kind of like an odd sock drawer category, I think. It's like, well, we don't know what it is. It's like black bile kind of thing. And so they used to think, well, people have disease, it's because they were out of balance, and we need to find a way to restore the balance. And a common way they would do that would be to take samples of blood. Oh, you're not feeling well? Let's just take a little blood from you. Okay, you'll feel much better after I take a pint, right? Might not seem to make a lot of sense right now, but it was an early attempt at some sort of scientific sort of approach to medicine. And this went on for a long time. George Washington, which this goes back to the founding of our country. George Washington really was still a proponent of sort of this theory of medicine here. So don't think this just goes back 2,000 years. This goes well into the history of our country here. So anyways, where's this whole conversation going? They call it humoral immunity, the ones that the antibodies produce, because these antibodies will make their way into all of your fluids and are good again for extracellular pathogens. Now we've already seen what an antigen is, are things that can generate an immune system response, are usually organic compounds that are found on cell membranes. Excuse me. If we talk about exogenous versus endogenous, exogenous means that they are found where? On the outside. Endogenous means that they are somewhere on the inside, and both of them can trigger an immune system response. They just do it slightly different ways. So this epitopes just refers to the different varieties of antigen receptors that you have here. Not going to test you on that. But this term, when they talk about naive T and B cells, when we say that the lymphocytes are naive, that's different than being immunocompetent. Do you remember that term that we used last week?
Yeah.
Immunocompetent just means that they have receptors on them.
Mm-hmm.
All right? If they've got receptors and are immunocompetent, as long as they have not come in contact with their particular antigen, they're naive. That's what it means. Means they're immunocompetent but have yet to come across their particular antigen. And you hope that that most will never get used, because you produce a huge variety of them. The way it works is just, an overview is just genetic recomposition. Now, what do I mean by this? Well, genetic recomposition. Imagine you've got a set of Legos. People know, I'm just talking about regular sets of Legos, all right? Not the special ones where you build a Star Wars Death Star or something like this that have special features. Just the regular sets of Legos. You can build a huge variety of different structures with just a couple of sets of Legos because of the way you just put the different pieces together in different ways. You get all these different shapes and forms from just recombining a couple of simple shapes in a lot of different ways. You produce an incredible variety of different structures from a small set of Legos. And it's the same with the way that your body produces these different receptors. You've got these different genes that code for parts of these antigenic receptors. And in the process of maturation, they just shuffle these genes around and say, I'm going to put a little bit of this protein, and then I'll put a little bit of this next to it, and then this one here. So you get this random assortment of pieces that end up being put together to form the antigen receptor. So that this is why you even only have a few hundred or thousand genes really for these antigen receptor proteins. You can produce hundreds of millions of different types of them just through this genetic recombination. Okay, so T cells. Let's do the T cells here. And we'll come back to this first one here where it says they have to recognize class 1 or class 2 MHCs. Here's the quick overview since I've got it up on the board here right now. Remember I said that the T lymphocytes have to have the antigen presented to them literally on a silver platter. Well, how do they do that? The way they do that is that there are these proteins that are called the MH1s 2 that act as fishermen. Now, what do I mean by that? Well, these are proteins that are produced inside and go fishing through the internal contents to pick up something. All right? They've got a little groove on them that allows them to pick up some cellular contents, and then they get taken to the surface to display whatever they have found. Look what I have found. I found this little protein here, or this little glucopolysaccharide, whatever it is. All right, that's how they work. They go fishing through the cell's interior, present it to the surface. Look what I found. If what they have picked up if they have been fishing through a healthy cell, they're going to pick up a healthy, normal protein that they'll be presenting on the surface. And basically that's saying, look, I'm part of you, I'm not a foreigner, I haven't been infected, and then the white blood cells should do what? Leave that cell alone. T cells won't activate it, hopefully, right? But if that's a cell that's been infected, or if it's a cell like a macrophage that has gone after a bacteria, that has engulfed it, digested it, now that little fisherman protein can display something that is foreign on the surface. And so that's what these MHC class 1 and class 2 are doing. They're the ones that are going to be involved in displaying the antigen that they have discovered inside the cell. So they have to recognize these fisherman proteins. We'll see them again later. But they also, in order to be effective, they have to not respond to your normal self-out. They have to be self-tolerant, or else you're going to start to develop an autoimmune disease. And we don't want that to happen. We don't want your body to attack your own cells. And this process of self-recognition, self-tolerance is occurring. If it's a T cell, what organ is this occurring inside of?
What was it that he just said?
Say it again?
What
What do you write-
organ
like, what
would
do you write
this
down?
development be occurring inside of if we're talking about a T cell here?
Never mind.
The thymus will be under the thymus hormones, yes. And so, sure enough, here's our picture of our thymus. And the T cells are developing that process. Now, I'm not going to test you on this little bit here, but the bottom line here, the bottom line is that during this maturation process, here's a thymic cell, here's a T cell. If the T cells have receptors on them that would be activated by your own proteins, that would be your own self-antigens, recognize your own self-antigens, that T cell gets destroyed. Kills. So it's a boot camp. It's a very tough boot camp. Right? Only those that can pass that test, that will not be activated by your own bodily proteins, that will be activated by something that's foreign, that undergo this positive selection, and those are the ones that get released into circulation. All that's occurring in the thymus gland, under control of all these thymic hormones here. Okay, so the T cells again have to be activated by sometimes presented to them on one of these two different types of showcase proteins, the MHC1s and the MHC2s. Now, there is a big difference in distribution in the body between the MHC1s and MHC2s. MHC class 1s are found on every cell with a nucleus, which means red blood cells are left out of the equation, but everything else will have MHC1s. That is different from the MHC class 2s. Class 2s are additional ones. To repeat, they are what? Additional ones that are found on lymphocytes and APCs. Who remembers what an APC is? An antigen presenting cell. So, would lymphocytes have MHC class 1s on them?
Yeah.
Yes. Would they also have class 2s?
Yes.
Yes. Keep in mind, this is an additional one that they have. So with the exception of red blood cells, everybody has class 1s. But the lymphocytes and APCs, aside from having class 1s, also have class 2s. And the important thing is that these two different types of MHC class proteins will be able to activate different types of T cells. you how this works. I'll come back. I want to show you this. We have, in order for them to be activated and be presented to them, they have to have a docking mechanism in place. And here's how this docking mechanism that will activate them works. If this is an antigen-presenting cell, and it is, will it have both class 1 and class 2 MHC proteins? And what they have on the surface here, they're on the surface, they're going to have a little groove on them. I need a different color, where they can present their antigen that they have discovered, or whatever they've discovered inside the cell. So here's the little antigen that they have picked up. Let's say they picked up the square virus. I hate that square virus. Now this has to dock in order to be activated with this docking mechanism called the CD8 cluster differentiation protein that will have a little place on it that has a receptor for the dreaded square virus here. So if the MHC1 docks with the CD8, they're only found on cytotoxic T cells, then we can activate a cytotoxic T cell. Can they kill directly the cytotoxic T cells?
Yes. Yes.
What cells in your body then have the ability then to activate a cytotoxic T cell? Which cells in your body at least have the potential for activating a cytotoxic T cell?
The helper? Helper?
Can red blood cells do this? No. No, because they don't have what?
So whatever have a nucleus.
Nucleus, which means they don't have the MHC class ones, right? So they won't be able to do that, right?
No.
Would epithelial cells be able to do this? Muscle, connective, neuron. Every other cell in your body has what? MHC1. So every other cell in your body technically has the potential for activating a cytotoxic T cell. This happens to be an APC in this example here, but this could have been an epithelial cell that would have a MHC1s, right? And that's a very common way of activating immune system response because the epithelial cells, they're lining these hostile, toxic environments where you're most likely going to be infected from, right? But the reason I put an APC here is because I want to show not only MHC1s, but also MHCs 2s. Because the MHC2s have to line up with the docking station that's called a CD, I can do this, the CD4 complex. The CD4 complex, we should show this up here. Here's my CD4 that can activate in conjunction with the MHC class 2s. And these CD4s are only found on helper T cells. CD4s are only found on what type of cells?
Helper.
Because they're only found on helper T cells, you often will use that term CD4 cell to refer to a helper T cell. So they talk about a population of CD4 cells, you know they're talking about which ones? Helper T cells. And the overwhelming majority of ones that are CD8 are the cytotoxic ones. Now, they have to be able to recognize both the CD4 have to bind with the MHC2, and the antigen has to match up with the antigen's receptor. Everything has to match up in order to fully activate. If we don't have all these things matching up, then nothing happens. But if all of these things line up, then we can activate our T cell. So as mentioned here, MHCs are found in what cells? Every cell that has a nucleus. And they're just showing how this fisherman protein works. I'm not going to go into details about it, but it's just trying to show, in this case here, we've got some antigen fragments here. Take it inside. Here's my class 1 molecule that has picked up this little bit here in the little groove here. And now we're going to drag it to the surface to present to the T cells to activate them. That's MHC1s. MHC2s, similar thing. They just activate or gain access to the antigen a slightly different way. They have to pass through a phagocyte this way. I'm not going to test you on this. But if this is an MHC class 2 molecule here that's now displayed on here, what kind of cell would this be here?
Helper T cell.
Well, this would be an antigen presenting cell. This could activate a helper T cell. So there's a helper T cell coming up down here, right? In your imagination, there is in my imagination, that's going to hopefully be able to be activated by this. But this could be an antigen presenting cell, because the antigen, or a lymphocyte, because they're the ones that have which ones? The MHC class 2s. All right, now B cells do the same. Let me just show you this. Let me just show that again. No. So the B cells, just to remind you something. Do the B cells need to have the antigen presenting with the MHC 1s or 2s? No. When you think MHC class 1s and 2s, and you think of CD4s and CD8s, you're not thinking about B cells at all, are you? You're just thinking about what cells? T cells. T cells have this. The MHC 1s have to dock with the CD8s. The MHC 2s have to dock with the CD4s. Stupid pet trick number 1. What's 2 times 4?
8.
What's 1 times 8?
8.
So no matter what you get, you have what equals to the numbers of fingers you have.
8.
Assuming that maybe you lost your thumbs in a farm accident or something. Yeah, whatever it takes. 2 times 4, 8. All right. 1 goes with 8. 2 goes with 4. So can the B cells, quick question, can the B cells get an initial activation? We'll see this in a slide. Can they get initially activated by a free-floating antigen? Just answer the question, yes or no?
No.
Yes.
B cells, can they get initially activated by a free-floating?
Yes.
Yes.
Yes. Everybody?
Yes.
Yes.
Can B cells get initially activated by a free-floating antigen?
Yes.
Yes. Do they need to have it presented to them?
No.
Can T cells be initially activated by a free-floating antigen?
No.
Do they have to have it presented to them?
Yes.
On a silver platter.
Thank you, exactly. Okay. So do the B cells develop their receptors? Do they develop immunocompetence? Do they develop their receptors? Do they develop it in the bone marrow or in the thymus gland?
Bone marrow.
In the bone marrow, hence the B for
Bone.
bone marrow here. And they go through the same, a similar kind of boot camp. In other words, they only get released to the circulation if they can recognize something foreign. So how are they going to get it, and how are we going to activate this immune system? Well, how are the antigens getting past? Well, somehow antigens have made it past your body's first line of defense. You've got a splinter, for example, that's been able to penetrate through your skin, or some issue in your GI, respiratory tract here. Common way that happens is they'll enter into your lymphatic system, get trapped maybe in the spleen, all right, or end up in lymph nodes, or in those mucosas associated lymphatic system, your tonsils, the Peyer's patches. And once they're there, right, then both the cellular and the humoral immunity is going to be activated in the following sort of 3 stages. Cellular and humoral immunity. Cellular, is that the T cell or the B cell response? Cellular immunity. T cell. Because they're the only ones that can directly attack cell, the cytotoxic T cells. Humoral immunity. What is that a function of? That's the B cells. B cells, humoral immunity, because their antibodies go into all the fluids. The T cells, a cellular response, because they're the only ones that can kill whole cells themselves. So we have to first recognize a specific antigen, undergo what's called clonal selection, where they'll start to multiply to become an army of clones that all can recognize and fight against the same antigen here. For T cell activation, we need co-stimulation, which means co-stimulation means we have to have all of this crap happening. The MHC1s have to activate the dock up with the CD, 8s like this. Then they actually have to stimulate each other with the right kind of cytokines. Like you release interleukin 1, and then she'll talk back with interleukin 2. And then that interleukin 1, interleukin 2 dialogue triggers them to start dividing into a massive army of clones. So they involve this clonal expansion. Just means once they've been activated, we're going to start to multiply, divide into an army of identical clones that will all be able to fight against that same antigen here. All right. So this little bullet point where it says, in general, when an antigen initially enters the body, there's only a small number of lymphocytes that have the correct type of receptors on it, the B cell receptor, the T cell receptor on it here. And that's why you need to undergo clonal expansion, all right? Make an army of clones, because it's just one activated cell going to do the job? No, you need an army of them. And then the last one here is that the last of the R's, the 3, recognize, react, remember. Remember means that some of the clones that are produced will not get involved in the initial fight, that they will produce memory cells that will hang back and then be able to mount secondary responses if you're activated in the future. Same response. So this is showing how a T cell would respond here. Make this a little bigger. This T cell is virtually identical to the antigen presenting cell, or this dendritic cell, I'm sorry, is similar to the antigen presenting cell. Is the dendritic cell an antigen presenting cell, by the way? Yes, all right. Except this picture is not nearly as pretty, of course, as the one that I drew. But leaving that aside here, they're trying to show the same things here. Is that T sub C is a way of talking about a cytotoxic T cell. And the cytotoxic T cells would have what kind of cluster of differentiation? As CD8. And that has to dock up and recognize the class 1, MHC class 1s, with its viral antigen, right? What if the antigen doesn't match the antigen receptor? Is anything going to happen?
Nope.
No. Just going to keep going. Just walk away. And we have the same thing over here, except over in this one, we've got the MHC class 2 that can dock up with the CD4. CD4s are found on which population of cells? Helper T cells. Are they found on B cells? No. What type of cells? Just the helper T cell. And then they will start to undergo this dialogue. They'll communicate with cytokines. That's that co-stimulator here they're talking about. And then the magic happens. That co-stimulation, everything works, it's going to start to trigger them to undergo massive growth and division. So the one cell divides to become 2, to become 4, to become 8. 5, 6 days later, now I have 1,000,000 copies of that one cell, 1,000,000 copies of that one clone here. Most of them are going to be involved in the initial fight, but some of them will hang back to become memory T cells. Memory T cells are the ones that will help to give you immunity down the road. How are we doing with time? For full activation, that initially activated cell has to get further feedback from an activated helper T cell. So even after we've activated a cytotoxic T cell, that activated cytotoxic T cell will not undergo full clonal expansion unless it's also been activated by a helper T cell. So without the helper T cell, we're not going to get proliferation of any of the other populations of cells. We're not going to get proliferation of the B cells, or the cytotoxic T cells, or any of the other ones here. And it's just trying to show that this cell is going to secrete cytokines like the interleukins that we talked about, which are going to be coordinating the immune system response. Will they coordinate some of the things that we've already spoken about? Do you think some of these will be involved in fever?
Yes.
Do you think some of these are going to be involved in causing B and T cell to proliferate, to divide over and over again? Yes. Do you think they'll enhance phagocytosis?
Yes.
All of those types of killing effects we're going to see. Am I going to expect you to memorize what interleukin 1 does versus interleukin 2 versus interleukin 3? No, 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 no. But you should know that interleukins, as the name implies, are chemicals generally released by activated white blood cells to trigger the activity of other white blood cells. And a critical group of cells that they're released from are the lymphocytes during the activation process. Can helper T cells kill?
No.
Can regulatory ones kill?
No.
Can cytotoxic T cells kill?
Yes.
They're the ones that can kill directly. And they primarily kill by lysis. they release compounds called perforins. Did I give you that name before, perforins? Maybe not. It's a great name, perforins. Because that name, when you perforate something, you're doing what? Poking holes in it. So perforins are a defensive protein that will poke holes in the target cells, causing them to do what? Life. There are other things that they can produce too. They produce compounds that will cause DNA in their target cells to get scrambled, to break it up. And of course, once you destroy the chromosomes in DNA in your target cell, have you effectively killed it? Yes. All right. Now B cells, and we'll have to finish up with this here. Can B cells be activated by just a free-floating antigen? Everybody should say? Yes. And that's what they're trying to sort of show here. So we are initially activating our B cell with just a free-floating little cone-shaped antigen here. All right? Will that initial activation be enough to get them to start to grow into an army? Or is it going to need some help from an additional? Going to need from which one? The helper T cell. And so that's why they're showing that lymphocytes also have to have this MHC, what, class 2 on them, right? Lymphocytes have these, and so these lymphocytes that have the class 2 on them here will then get fully activated by a helper T cell. Here's a helper T cell. And again, co-stimulation, interleukin 1, interleukin 2. And once they've had that feedback from the helper T cell, now the B cell will start to do what? Divide. Undergo massive rounds of cell division. Will all the B cells be involved in the initial fight? Will some of them hang back to become memory cells? Yes. Some will become memory B cells. The ones that are going to be involved in the initial fight, mature into these antibody factories that we call a plasma cell. And the plasma cells just hang out in your spleen, your lymph nodes, your tonsils, wherever like this, just churning out massive quantities of what? Antibodies. Antibodies of a wide variety of different shapes and sizes. And I think we'll have time just to finish talking a little bit antibodies. Since we've talked about this before. I've seen this in the lab a little bit here. Antibodies, immunoglobulins, have this basic sort of Y-shaped structure. So which is why they always tend to draw them with a Y. I'm not going to be that very much concerned about the pair of heavy chains versus light chains, but what is important to them are these two different colored areas here. The orange ones are called the constant regions, and the green one up there are called the variable regions or variable segments. It is the constant segment that determines what class of antibody we're dealing with and what kind of powers it has. Whereas the variable region, the variable segment, is what makes it antigen specific. Let me repeat those two things. The constant segments, the constant regions determine the class. We'll see what that means in a minute. That means whether we're talking about IgA, IgG, IgE, IgD. But it is the variable segment that determines its antigen specificity. What kind of antigen it can mount a response to. So here are our different classes of antibodies. Starting with this simple little Y-shaped one that's called IgG. IgG is far and away the most abundant far and away it's the most abundant. It's almost all you get in secondary infections. It's a primary thing you get during secondaries. It's the only one that crosses the placenta. It's the only one that does what? Crosses the placenta. One thing that I was happy about on the lab midterm is that almost everybody got that question about what kind of RH issues would affect mother and child. Almost everybody got that one correct. Even without the whole erythroblastosis fetalis, understanding that it was RH negative women who are having RH positive babies. That's where the issue comes into play. It's the second one that becomes a problem. Almost everybody had that right. A few people had some issues with that. But it's IgG. What we're talking about, the ones that can cross the placenta and cause that, is IgG. It's the only one that crosses the placenta. Now, IgA is interesting. You produce tons of it, but not inside of your body. It's on the surface of your bodies. So you produce tons of it, but in your secretions. So you find this in all your mucus secretions. In tears, you find some in saliva, in breast milk. I'll focus on this one and we'll finish them up. IgM is this massive big pentamer. Big parts here. It's the first class that's released in a primary response. So when you first begin to get sick with something, the plasma cells first begin producing IgM, but then later start producing more IgG. IgM is the first class that's released. It's the first antibody secreted following the initial exposure. I mentioned in the lab that this is the one that's produced in response to foreign ABO blood types. Way too big to cross the placenta. This is why you're okay if you're a mom who has type A blood, carrying a baby who could be B, because your anti-B IgMs will never cross the placenta, way too big. IgE, E stands for evil. Because IgE is it's responsible for anaphylactic shock, for allergic responses, binds to allergens. They're the ones that trigger the mast cells to initiate this explosion of inflammatory chemicals. IgA is evil. All right, let me finish up with this. IgD, I'm not even testing on because IgD technically is an antibody, but it's really just the B cell receptor. Turns out the reason that we draw B receptors looking like, when I was drawing Sue the other day with her little receptor that looked like this, the reason we draw them look like this, a little Y shape, it's an antibody. Technically, it's an IgD. Now, before you put stuff away, quick question here. 2 lymphocytes here. This will be Dave. And this is Stan. Dave can recognize, let's say, swine flu. And let's say Stan, I don't know, the tuberculosis. Well, whatever, okay. So when you're first exposed to something, the first class you can produce is what? IG. And then later, you'll start to turn over and start to produce more of what? IgG. Stan would do the same, right? IgM. And then later produce what? IgG. So I want you to compare this antibody and this antibody here. All right? Are the constant regions the same?
Yes.
Are the constant regions on these the same?
Yes.
Yes.
No.
Just some of the antibodies.
No, because the constant regions determine what class we're in, right? This is what, IgM, this is IgG. So are their constant regions the same? No. Are their variable regions the same?
Yes.
Yes. Their variable regions are the same because both of their variable regions would be able to recognize, in this case here, swine flu here. Would it be the same with Stan's over here? Would it be the same? But I want you to compare Dave's IgM with Stan's IgM. Do these two have the same constant regions?
Yes.
Yes, because they're both IgM. Do these two have the same variable region?
Yes.
Why not?
Because it's two different,
Two different antigens, right?
Yeah.
Different. Would it be the same with these two here? Would these two share the same constant regions?
Yes.
Because they're both what? IgG. Those are the constant regions the same. Are their variable regions the same with these two?
No.
All right. That's going to be it for today. I'm going to let you go a little